The overall goal of this experimental procedure is to map the spatio-temporal gene expression dynamics of the vertebrate segmentation clock using fixed tissue sampling.
This method allows an unbiased assessment of oscillatory gene dynamics in the pre-somitic mesoderm, and this is crucial to understanding the molecular mechanisms governing vertebrate somatogenesis.
The main advantage of this technique is that many samples can be generated and processed simultaneously, allowing the high-throughput analysis of gene expression dynamics in fixed tissue.
Demonstrating the procedure will be Dr. Charlotte Bailey, who's a former postdoc in my laboratory.
After obtaining a mouse uterine horn according to the text protocol, under a stereo microscope, use curved scissors to cut the thick muscular membrane of the uterine horn and carefully extract each embryo. With curved scissors and fine forceps, dissect away the amniotic sac from each embryo. Then, using either a surgical needle or curved scissors, harvest the tail tissue from each embryo by cutting the embryo anterior to the rear limb buds. Balance the tail tissue ventral side down. Then generate pairs of PSM explants by dissecting the tail tissue into two halves along the midline, using a gentle rocking motion with the needle. Ensure that the neural tube, notochord, and PSM tissue are equally divided between the two explants. Then pipette each contralateral PSM explant onto the underside of a 35 mm plastic culture dish lid in a small volume of pre-warmed culture medium. Place the dish on the